All right, we're taking a look at cellular respiration part 2. We're going to start with the second major stage of cellular respiration, which is referred to as pyruvate oxidation, sometimes called the intermediate reaction. Where we left off in our last video was at the end of glycolysis. Glycolysis represents the first primary stage of cellular respiration, in which glucose is broken down or oxidized through a series of 10 steps, ultimately to produce pyruvate. Pyruvate then is a organic 3-carbon compound left over from the remaining carbon atoms that were originally in the 6-carbon glucose. Okay, so we have now 2 pyruvates. All 6 carbons are left from our glucose, but they're now in 2 separate molecules of pyruvate, each one with 3 carbons. So in glycolysis, we took a bunch of the electrons and some of the hydrogens off, rearranged the atomic structure, generated ATP through phosphorylation, and now we've got all this leftover organic matter. In part 3, we'll take a look at what happens with other types of reactions outside of cellular respiration. For example, fermentation reactions. But in the presence of oxygen, with cellular respiration being aerobic, what will happen to the remaining carbon is it will start to be fully broken down. So rather than having some carbon left over, we're going to fully oxidize that so that there's nothing left, and the cell is going to strip all the remaining energy from it. Okay, so that starts in the next stage here with pyruvate oxidation. Oxidation being the breakdown or loss of electrons, loss of hydrogen from pyruvate. Now, from the perspective of the cell location, glycolysis took place in the cytoplasm, and now we're going to see the remaining stages move into the mitochondria. So inside the mitochondria, we're going to see the breakdown of pyruvate. We're also going to see the breakdown via the Krebs cycle, and ultimately the electron transport chain. So since we're moving into the mitochondria, let's just take a quick look at some of the structural aspects, some of the different regions. There are 4 distinct regions of a mitochondria. The first is called the outer membrane, the very sort of outer layer. The next is called the inner membrane space. That is the space in between the outer membrane and then what's referred to as the inner membrane. So you've essentially got 2 membranes, the outer and the inner. And then the inner membrane space is the region in between those two. And then finally, what's called the mitochondrial matrix is the space in the very middle. So 4 areas. We're going to look, all of these actually are going to be key when we look at the electron transport chain. Pyruvate oxidation, as well as our next step, the citric acid cycle or the Krebs cycle, are going to take place inside the middle here of the mitochondrial matrix. So the mitochondria is going to have to import pyruvate into the mitochondrial space, the matrix, and it's actually going to use some energy with that. It's actually going to use some ATP to pump that in there. But the matrix then is going to be where these remaining steps take place, or the next step, excuse me. So pyruvate gets pumped in to the matrix. It's got 3 carbons. And what's going to happen in the pyruvate oxidation is it's going to be oxidized, meaning it's going to lose electrons, lose hydrogen. And what's going to happen is it's going to be broken down to form a 2-carbon molecule called an acetyl group. So we essentially go from a 3-carbon pyruvate to a 2-carbon acetyl group. Now, in the process of that breakdown, we're going to have the removal of hydrogen from our pyruvate, and that hydrogen is going to be added to NAD, our electron carrier, which is going to be reduced then to NADH. So this actually marks the first step in which the carbon atoms are going to actually be lost to the environment. So from the first stage of glycolysis, we haven't actually lost any carbon up until now. We've rearranged it, we've lost some hydrogen, electrons, we've added and removed phosphates, but we haven't actually lost any of the core organic material, being the carbon. But that's going to change now. So what's going to happen is called a decarboxylation reaction. Okay, decarboxylate simply means to remove carbon. Essentially is what that means. Now, when a carbon atom is removed, one of the first things that's going to happen is it's going to react with O2 in the air, O2 being nearly ubiquitous, being everywhere. So as soon as that carbon is pulled off, it's going to react with O2, and it's going to immediately form a more stable compound, which is CO2. So ultimately, the removal of carbon, a decarboxylation, results in the formation of a CO2 molecule. And CO2 then is lost to the environment. It essentially becomes a waste product, and it's lost from any sort of meaningful use in the cell. Again, essentially becoming waste. So it doesn't actually go anywhere. The first law of thermodynamics states we can't actually lose it, but it's transformed into an unusable form, i.e. waste. So that's where our first CO2 is lost. Now, this is going to happen twice per glucose. So remember that one glucose enters, 2 pyruvate molecules are formed per glucose. Each one is going to be decarboxylated via this reaction here. And so we're essentially going to lose 2 carbons per glucose at this step. So we're losing carbon, we're gaining a reduced form of NADH. Now, one other interesting thing that happens here is the 2-carbon compound is too small, apparently, to function on its own. And so this is somewhat of a random thing, but this is what happens in nature. So this 2-carbon molecule has to actually be picked up and carried by what's called a coenzyme. And the name of this coenzyme is called coenzyme A. CoA is the abbreviation here. So the acetyl group, the 2-carbon molecule, gets picked up by a coenzyme carrier and forms a compound called acetyl coenzyme A. And this now becomes the group that's going to enter into the next stage, which is the citric acid cycle or the Krebs cycle. Okay, so that's called the pyruvate oxidation, also known as the intermediate reaction. Now, acetyl coenzyme A is going to enter into the citric acid cycle, the Krebs cycle, where it's going to form with a compound called oxaloacetate. And we're going to see that here in just a second. Let's recap what happened in pyruvate oxidation. Per glucose, 2 carbon dioxide molecules are produced, 2 NADH are generated, 2 acetyl coenzyme A complexes are created. Now, for each glucose, 2 of these acetyl groups are going to enter into the citric acid cycle. So the citric acid cycle, also known as the Krebs cycle, is considered a cyclic metabolic pathway. The reason it's considered cyclic is not because the compounds literally form a circle in the cell. The reason it's considered cyclic is because the starting compound is transformed and regenerated at the last stage of the cycle. So, for example, in this case, we start with oxaloacetate, and oxaloacetate reacts with our incoming product here, acetyl coenzyme A. These two are going to react to form a new product called citrate. Citrate then becomes our product in the first step of the reaction. The remaining steps are going to transform citrate, further decarboxylate, remove the carbon, and ultimately we're going to end up back where we started with oxaloacetate. And with the new acetyl coenzyme A coming in, we'll start over again. So the process is considered cyclic because it regenerates the starting compound so the process can take place again. This also takes place inside the mitochondrial matrix, just like our pyruvate oxidation. So same location here. Now, as I just mentioned, oxaloacetate is going to form to make citrate, in which case then it's going to be transformed. The primary products of the citric acid cycle are reduced electron carriers, NADH and FADH. So the starting unreduced forms, NAD and FAD, are going to be reduced via the cycle, via the citric acid cycle, and we're going to have reduced forms of NADH and FADH respectively. We're also going to produce a little bit of ATP, similar to glycolysis. We actually do produce a little bit of net ATP here, but ultimately that amount is somewhat trivial. It's not a high yield. So even though we do actually produce a little bit of ATP, the primary takeaway from the citric acid cycle is a large number of reduced electron carriers. And in the last stage, we'll see how they play a role in the electron transport chain. Okay, so next we're going to walk through the steps of the citric acid cycle. So there's 8 steps in the cycle. The first step here, we see that the incoming acetyl coenzyme A, carrying our 2 carbons left over from glycolysis and the pyruvate oxidation, they're going to immediately be joined together with oxaloacetate, the compound that kind of begins this process. Oxaloacetate is 4 carbons, acetyl is 2. These are joined together to make a 6-carbon compound known as citrate. And citrate is largely what the cycle is named after, the citric acid cycle. A lot of places, a lot of resources refer to it as the Krebs cycle, which was after Dr. Krebs, who discovered the cycle. So the citric acid term is a more practical name based on the actual molecules. The Krebs cycle is more of a tribute to the discoverer himself. Anyway, we now have a six carbon compound known as citrate. The enzyme here is known as citrate synthase because it synthesizes citrate in this reaction. And in the process, the coenzyme carrier is actually released, and it can actually be used again in a different reaction later, so it can be recycled. Also, a water molecule contributes oxygen in this reaction, so we're adding some oxygen in the process. But ultimately, this is a key synthesis reaction in which our carbon comes in, joins to the oxaloacetate to make a 6-carbon compound. Now, with the remaining steps, we're essentially going to see the transformation of that carbon through different stages. Starting with the next step is an isomerization. If you recall from our first video, an isomerization is really just a rearrangement of the carbon and the oxygen and the hydrogen. This enzyme is called aconitase, excuse me. I'm not sure if I'm saying that right. Aconitase, I believe, is right. Step 3 is a key reaction in which we're going to have another reaction that generates NADH, called a dehydrogenase. What I meant to say there was another decarboxylation. Also a dehydrogenation, but a decarboxylation was the word I was looking for there. So in other words, we're going to remove another CO2, another carbon in the form of CO2, and what we're going to see here in step 3 is that another carbon that started out in the form of glucose is now going to be removed. So we're actually going to be down to 2 carbons left from glucose after this one is removed. Okay. In that process of decarboxylation, we're going to charge up another NAD to NADH in the process. We now have a 5-carbon compound, and its name is called alpha ketoglutarate. And the enzyme is called isocitrate dehydrogenase. Step 4, we're going to lose that remaining carbon, and we're going to be completely stripped of the carbon originally found in glucose. So therefore, another decarboxylation reaction. We're going to take alpha ketoglutarate and transform that into a compound known as succinyl coenzyme A. So our coenzyme carrier is going to come back and make another appearance here, carry this molecule to the next step in the process. Okay, so it now becomes succinyl coenzyme A. CO2 is once again lost to the environment. Our remaining carbon is gone. Another NADH is generated. The enzyme here is called alpha ketoglutarate dehydrogenase. Just to recap real quick where we're at, we brought in 2 carbons following the pyruvate oxidation, what we had left from glucose. We've essentially lost both of those, one here, one here. All the carbon is now gone. All the carbon that entered this first stage of glycolysis, it's now lost to the environment as CO2. It's a waste product. What we ultimately have left now are some of the electrons and hydrogens and the energy that was transformed from that initial compound. Also, some of it's been added from water. Some of it gets added via phosphates. But ultimately, the carbon is gone. What we have left is the potential energy. And some of the remaining steps of the cycle are going to transform that potential energy into more electron carriers, and we're going to generate a little bit of ATP here. So in step 5, we generate some of that ATP. It actually starts out as a molecule called GTP, guanine triphosphate, but GTP is transformed shortly thereafter into ATP. So ultimately, the net result is a formation of ATP. So our enzyme, our compound succinyl coenzyme A with 4 carbons is transformed into what's called succinate. The coenzyme carrier is kicked off once again, and we get a little bit of ATP via substrate level phosphorylation produced here. If you forgot, or if you're watching this video first, substrate level phosphorylation was talked about in the part 1 video. Okay, so we've lost the carbon, we've generated a couple NADH, and we just generated ATP. And that's going to happen twice for each glucose. In the next stage, this is where we actually generate the other electron carrier, FADH2. This is actually the only stage in which that takes place. So succinate is going to be acted upon by an enzyme called succinate dehydrogenase, which is going to strip hydrogen from the compound, transfer those to the carrier FAD, and generate FADH2. This one carries an extra hydrogen, therefore it's referred to with the 2, FADH2. So this is going to charge up our electron carrier here. The next stage is kind of a reformation of the previous molecule. Fumarate is transformed into malate. Water plays a role here. And then the final stage is where malate is going to be transformed back into the starting compound, oxaloacetate. So this is the step that makes it cyclic, kind of completes the cycle. So our enzyme is called malate dehydrogenase. It is going to, once again, take a hydrogen and add that to NAD, creating NADH. And as I mentioned, regenerate our starting compound, oxaloacetate. And now our process is complete. Oxaloacetate can now cycle back, reform with acetyl coenzyme A, and start a new process, assuming more acetyl coenzyme A is available to join. Let's take a look at some of our yields here. So from the citric acid cycle, we produced per glucose 6 NADH, 2 FADH2, 2 ATP, and 4 CO2. Going back here, per acetyl coenzyme A that enters, we generate 1, 2, 3 NADH. Since we have 2 per glucose, we multiply that by 2. So we get 2, 4, and 6 NADH. Looking at our CO2, we get 1, 2 CO2, double that to make 4. We get our 1 ATP here, double that to make 2. And we get our 1 FADH here, double that to make 2. So that's where we come up with these numbers here with our yields per glucose. And as I already mentioned, all the carbon's been lost. What we have left is a little bit of ATP, but mostly we have charged electron carriers in the form of NADH and FADH2. If you've ever wondered where most of your food actually goes outside of the waste that we expel when we go to the bathroom, where most of your energy and the organic matter itself actually goes is right out your lungs. So most of your breakfast, for example, or your lunch or dinner, you'll actually exhale it in the form of CO2. So you're actually exhaling the majority of your organic matter through the lungs, and that's actually where it goes. Some of it obviously will go through different orifices in the body, but a large amount of it actually is breathed out of the body as CO2, and that's a major component of our food waste, CO2. So we've essentially transformed the majority of the organic matter into waste as CO2 and potential energy in the form of NADH, FADH2, and we've produced a little bit of ATP. The amount of ATP we've produced so far has been somewhat minimal compared to what we will produce. We generate about 88% of the energy per glucose in the very last stage of the process, known as the electron transport chain. So we've produced just over 10%, about 12% of the potential energy in ATP so far, but most of that energy actually exists in these electron carriers. So they're like little batteries, so to speak, and they're carrying a charge that will actually be used to generate the other 88% of the ATP that has yet to be created. And that generation of that remaining ATP is going to take place here in the electron transport chain. Now, this takes place in what's called the inner membrane of the mitochondria. So this little spot right here is called the inner membrane. So you got the outer membrane, the inner membrane. The other cycles, pyruvate oxidation and the citric acid cycle, were taking place inside the matrix. But now we're moving to the membrane itself. So from the space to the sort of the wall, so to speak, here on the inside. And if we look at that, I'll show you this figure here several more times. But if we look at here real quick, this phospholipid layer is that inner membrane here. So this is a similar compound that we have in a cell membrane, a phospholipid bilayer. Embedded in that membrane, in many instances, lots of repeating units of this, you have a series of 5 enzyme complexes referred to in the next part here. I'll list those out here. But the electron transport chain takes place here in the inner membrane via a series of 5 major complexes that are going to take this potential energy and transform it into ATP. And that's what we're looking at lastly here in part 2. Now, it's a pretty amazing process, in my humble opinion, what happens here. And it's no short of extremely complex. So we're going to look at it from a broad concept without all the nuts and bolts and the details behind it. But to even begin to understand what's really happening here, you have to recognize that there's really 2 different forces at play in order to generate ATP via this electron transport chain. And one of them is referred to as chemiosmosis. Chemiosmosis. And if you remember, osmosis is the movement of water across a membrane. Chemi is in reference to chemicals. So the movement, in this case, of chemicals, or ions specifically, across a membrane is what chemiosmosis means. That is going to involve ions known as H+, hydrogen ions, and they're going to move across the membrane. So chemiosmosis involves the establishment of an H+ gradient. I'll explain that further. That's the first step. It's essentially building up pressure in the form of a chemical. Next, then, that pressure of that chemical gradient is going to activate this pretty amazing enzyme called ATP synthase. ATP synthase essentially does all the work. This thing right here is one of the wonders of the world, in my opinion. A very amazing little enzyme complex that generates ATP by being fueled by chemiosmosis. So chemiosmosis is like the gasoline to your car engine. And the car engine really does all the work, but it needs a little bit of fuel in order to drive. And so that's kind of like what chemiosmosis is. So if you were describing how your car works in a very broad sense, again, not looking at every single part of it, like in a really broad sense, you'd have to say something like, well, you got to put gas in it and then hit the pedal. And that's kind of like what we're saying there. Chemiosmosis is the gas, ATP synthase is the car. So ATP synthase, in a simplified explanation, is going to take ADP that is generated in the cell, phosphate, which is going to come from our environment, and couple these two together or join the two together to make ATP. Now that's an endergonic reaction, so it's essentially like going uphill. And so it's going to need an energy gradient in order to drive that reaction forward. And that is where our chemical gradient from chemiosmosis comes from. Chemiosmosis essentially begins when a high concentration of hydrogen ions are established on one side of the membrane. Backing up here really quickly, this is going to take place, this concentration of hydrogen is going to take place in this little area here called the inner membrane space. In between the inner membrane and the outer membrane, there's a space here called the inner membrane space. This is where the high ion concentration is going to be generated. So a high concentration here means you have pressure. And what will happen is when the pressure of these chemicals is built up, they'll want to move from a high area, from an area of high pressure to an area of low pressure, or in this case, concentration. So through diffusion, the movement of particles from high to low, these chemicals will want to move in response to that gradient. The problem is that they can only move through areas where they're allowed to migrate. So it's kind of like having water trapped inside of a vesicle. The water would move out of it if you open the vesicle, but it's trapped inside, like a water bottle. So what happens in the case of ATP synthase is it acts as a valve to allow the hydrogen, this concentration, to move through it, which is like the equivalent of taking the cap off of a water bottle. Okay, so imagine you have a water bottle trapping the water inside. The water would easily move out of it if you open the cap and turn it upside down, but as long as the cap's on, it remains inside. ATP synthase acts like a valve or an opening for these molecules to move through, like a water bottle has its own cap on it. Okay, and so that movement here is referred to as chemiosmosis, the movement of chemicals or ions across a membrane down their concentration gradient, meaning from a high concentration to a low concentration. A way to think about this is this works a lot like a water dam generates electricity. Just to give you a quick visual, I pulled a figure off the web real fast here. A water dam generates hydroelectricity, in which you have a lake behind the dam back here. That's like what you would see back here. This is essentially pressure being built up. So this water represents a lot of potential energy in the form of pressure, like air inside a balloon. Okay? And so here's that pressure. As it moves down a release valve, that potential energy actually moves through and drives a turbine that creates friction, which is transformed into electricity, which is then transmitted through power lines and then ultimately to its destination. It may go straight to your house, for example. Probably not straight to your house, but it goes through a series of grids before it ultimately is delivered to wherever it's going. the reason I mention all that is because this process here is a lot like that. You have a concentration, in this case rather than water, of ions, and like moving through that release valve in the dam, they're going to move through this little area here, which happens to be through the ATP synthase complex. So this acts kind of like that valve for the water turbine to allow these chemicals through. And as these chemicals move through this opening here, it actually drives the formation of this rotor complex here. This is in the ATP synthase. And the movement of that rotor complex will actually couple the formation of ATP, joining ADP and P phosphate together. Now, that's not going to make perfect sense. There are some really cool videos online. I'm not going to show all those because it's something that's fairly complex and it gets down to some fairly specific details. But if you'd like to see some of that, just type in ATP synthase animation on YouTube and it'll show you that. But ultimately, what you need to know for this class, the concentration drives the formation of this rotor, and that creates ATP by joining these two together here. Now, to understand then how that concentration gradient is established, this is where the electron transport chain comes in. Okay, so just to reiterate, here's something we talked about: chemiosmosis and the establishment of an H+ gradient, and then activation of the ATP synthase enzyme. So in order to establish that concentration of ions, this is where the function of our electron carriers and the electron transport chain itself come into play. So they are, as a series of steps, they're going to actually pump these hydrogen ions into this space here that will actually create the high concentration, which will then generate the gradient to move through the ATP synthase complex to activate the enzyme to produce ATP. So, as it says down here, the H+ ions are trapped, and the path of least resistance for them to exit is actually through the ATP synthase enzyme. So here's a figure showing the enzyme complexes involved in that process. So what's going to happen is the electron carriers, NADH and FADH2, they're actually going to move right up next to these complexes that are established in the intermitochondrial membrane, and they're going to transfer their potential energy to these complexes, which is like the equivalent of hooking up a battery to an electronic device. Once the battery is hooked up, inserted, it can transfer its energy to the device, and the device can become activated and do whatever it does. That's essentially what's happening here. These NADH and FADH molecules have potential energy like a battery, and they're going to get hooked up to this complex here. And when they get hooked up, what's going to happen is they're going to activate the complex. Now, what the complex does is it takes the hydrogen ions that are found naturally within a cell. These are ubiquitous little particles, so they're found anywhere a cell would be. They're partly generated by the cells themselves. They're also just part of the environment. So to clarify, they're kind of like water and oxygen. These things are just everywhere. So they're present, and the activation by NADH here is actually going to cause this H+ ion one at a time to actually be picked up and pumped through this complex into the intermembrane space of the mitochondria. Let's go back one more time and just look at this figure here real quick. So the okay, I wanted to find just a little better picture so we can see this from a more flat perspective. So we're here in this inner mitochondrial membrane, and it goes all around the mitochondria, and in different locations throughout we'll have repeating series of these electron transport chains here. Looking at these here, these are more detailed more detailed look at those there. So that's what we're seeing here in the series. What's going to happen then is they're going to be pumping hydrogens from here. This is the matrix in this area here with the slightly darker gray color, pumping hydrogens into this space here with the teal color here, which is the intermembrane space. And this is ultimately then where our concentration gradient is going to start to take place. So the concentration gradient here is taking place in that turquoise area. Okay, so we're establishing what I talked about here. So what actually generates the gradient is the activation of these complexes by NADH and FADH respectively. What's really interesting is that the primary means by which your food generates energy for you is by actually moving hydrogen ions across a membrane. It's a really complex way of doing a very simple thing. Your body goes through all these elaborate processes, breaking down your food into a multitude of steps with very complex chemical reactions that all ultimately result, most of that work, I should say, results in the movement of this little thing here. Okay? And that's really what's happening here. It's kind of an amazing series of events for something so simple. But this doesn't just move on its own. It takes energy, and the energy comes from the NADH, which comes from your food. So now, what we call these complexes here, there's actually 4 complexes, and then the 5th part of the chain is the ATP synthase. So there's I said it back here. So, I'm sorry, I'm going all over the place. Apologize here. It's on this next one here. Sorry, I was getting ahead of myself. There are 5 complexes within the chain. 5 components, I should say. 4 complexes, and then the ATP synthase enzyme is number 5. So of these 4 complexes here, 3 of these are actually going to be pumping hydrogens across. Another one just plays a role in a slightly different way. So you have actually what are referred to as proton pumps. The proton pumps take these protons. An H+ ion is a proton. It's a positively charged particle. And it's going to pump that into this space here. Okay? So that's kind of what reiterating here. This is a kind of a shadow figure of the last figure that we saw, taking out all the 3-dimensional aspects and showing you just a flat 2-dimensional figure here. Okay, so NADH is going to activate the first complex in the chain, and the activation of this first complex by NADH is going to lead to the pumping of 1 hydrogen across the membrane. The electron from this NADH then is going to be transmitted, follow this orange arrow here, is going to be transmitted from the first complex through some carriers to the next complex, following on to the 3rd complex, ultimately to the 4th complex, and then is finally coupled to oxygen, leading to the formation of water. Oxygen acts as our final electron acceptor. So it's like a little electrical wire. The energy simply moves from one area, the electron, all the way down this chain of events, ultimately then being dumped off to oxygen. And this is why you need to breathe oxygen, because oxygen will actually receive this little energy unit, the electron, so that it doesn't build up and cause harm inside the cell. So NADH will activate the first complex, and as that energy from the electron activates the first, it'll actually go on to activate the second, the third, the fourth, and then the energy will be passed to oxygen. FADH bypasses the first complex. It interacts directly with the second complex and passes its energy from the second complex onto the third, the fourth, and then to oxygen. So the electrons from NADH and FADH respectively will enter into complex 1 and 2. That's going to have a consequence here that we'll talk about shortly. Complexes 1 through 4, as I mentioned, make up the actual electron transport chain. So the transportation of electrons in a chain from 1, 2, 3 to 4, ultimately being passed to oxygen. The fifth part of the chain is the ATP synthase, which, as we've already talked about, will actually make the ATP. It is complexes 1 and 2 that actually receive the electrons from NADH and FADH respectively. Now, what's important to note is that of the 4 complexes, 1, 2, 3, and 4, only 3 of these actually act as proton pumps that move protons from one side to the other. And these are complexes 1, 3, and 4. What is going to happen with these pumps here is that they are going to move hydrogen across the membrane when activated. Now, the first pump will act with NADH, and so NADH will activate the first pump. That one activation will then trigger a reaction in the third and the fourth. So an NADH molecule will activate all 3 pumps in the chain. So one NADH will be responsible for moving 1, 2, 3 hydrogens across the membrane here. Complex 2 is what interacts with FADH. So when FADH interacts, it interacts here at complex 2, but actually bypasses complex 1. So FADH2 will only generate the formation, or the pumping, of 2 hydrogens, 1, 2 across the membrane here. This figure is a little confusing, by the way. It's showing what it needs to show is 1, 2, 3 hydrogens being pumped. So it looks like 4 here. It's really not. It's only 3. So FADH is going to interact with the second complex, not the first. Okay, and I'm going to may not make sense why I'm indicating that. It's somewhat of a random thing, but that happens. Okay, so I'm going to talk about that here on the next part, why that's important. For each for each H plus that we pump into this space, backing up one more time, for each H plus that we pump into this space here, an H plus will move through the ATP synthase enzyme and be responsible for generating 1 ATP. So for every H plus we pump into the space, we can theoretically generate 1 ATP. So 1 H plus equals 1 ATP in the grand scheme of things. It's what we call the theoretical yield. It turns out it's not quite that high, but it's the starting point for really understanding it, and that's all we're going to focus on. So it's what we call the maximum potential yield. Okay. So all we need to know for this class is 1 H plus equals 1 ATP. So what that means then is that the more, backing up one more time here, the more H plus you pump into this space, the more ATP you generate. Okay? Now, NADH will pump 3 hydrogens because it interacts with all 3 pumps. FADH will only pump 2 hydrogens because it only interacts with 2 of the 3 hydrogen pumps, being complexes 3 and 4. So what that means then is every NADH generated throughout cellular respiration will be responsible for generating 3 ATP. The FADH that we generate will only be responsible for generating 2 ATP, since they only pump 2 hydrogens per molecule, because they bypass the first complex. It's confusing. Let's be honest. It's all confusing. Cellular respiration is not an intuitive thing. It's very confusing. But just to reiterate here, for each hydrogen that we pump, we get to make 1 ATP. We charged up these electron carriers for the sole purpose of making ATP. So when we look at how effective they are, it's important to note how much hydrogen they're pumping, because that's what makes the ATP. So the NADH will pump 1, 2, 3 hydrogens, whereas the FADH will only pump 1, 2 hydrogens. And that's going to change the way we calculate our yield. So when we start to look at how much ATP we're getting per glucose, we go back and we look at all the stages of respiration. So in glycolysis, going all the way back to what we started in part 1, in glycolysis, if you recall, we produced 2 NADH per glucose. Pyruvate oxidation gave us 2 NADH per glucose. The citric acid cycle gave us 6 NADH and 2 FADH2 per glucose. So from the start to the finish, we had a total of 10 NADH and 2 FADH2. Now, calculating the totals then, in addition to the NADH, we had 2 ATP from glycolysis and 2 ATP from citric acid cycle. So we generated 4 ATP per glucose up until the electron transport chain, and then with the electron transport chain, we're going to see how we produce even more of that ATP. Okay, so easy math here, but just to show, we produce 10 NADH. Each NADH will pump 3 hydrogen, so 3 hydrogen per NADH. And as we talked about, one hydrogen equals 1 ATP. So if we have 10 NADH, each pumps 3 hydrogens. Each hydrogen gives us 1 ATP. That means we get 30 ATP for each 30 ATP per glucose out of the NADH that were generated. The FADH2, they generate only 2 ATP per glucose, so we get a total of 4 ATP from that. So our electron carriers gave us a total of 34 ATP. If we then add up the 2 ATP generated in glycolysis and generated in the Krebs cycle or the citric acid cycle, we would add another 4, giving us what's called the maximum theoretical yield of 38 ATP. The reason it's called the maximum theoretical yield, as I mentioned, is this is the greatest amount you could produce. The truth is, in reality, you tend to use more ATP that we didn't quite talk about, and you don't necessarily get to produce exactly 1 ATP per H+. So it ends up being closer to about 32 ATP per glucose, but the maximum theoretical yield that you could generate is 38. Okay? Okay, that's the last part of part 2 here. In part 3, we'll start with, as I refer to here, as metabolic integration, talking about how different molecules like proteins and fats and even different types of sugars can all integrate into the cellular respiration pathway differently, and how that gives us energy in different ways rather than starting with glucose. So we'll pick up on this next time, and we'll finish up respiration here with part 3.